Good morning, everyone. Welcome back to your channel, Medical Microbites. Extremely sorry for uh, delay in posting new topics. So today I'm here with the topic of overview of bacterial endospore. I have made this PPT for your understanding, knowledge, information, and so that you can write notes on this endospore sporulation, etc. So, what is sporulation in bacteria? It is the survival mechanism in adverse conditions. So, endospores are basically the doomsday preppers. Whenever the world or the environment surrounding the bacteria, vegetative form of bacteria, goes bad, no food, harsh weather, they pack all their essentials into a tiny, indestructible bunker that is the endospore. So, this is the sporulation. Now, definition of sporulation. This is a highly regulated process. First point is this is very much regulated. I will come to the points of regulations later on. This is a highly regulated multi-stage differentiation process by which certain bacteria, primarily within the phylum Firmicutes, that is the Bacillus and Clostridium species, transit from a metabolically active vegetative state to a dormant, highly resistant endospores in response to environmental stressors such as nutrient depletion or extreme conditions. So this is the differentiation process by which it transits into endospore, that is the highly resistant or dormant form, resistant to harsh weather conditions, so that it can survive in presence of environmental stressors, may it be nutrient depletion or extreme conditions, and transition from metabolically active vegetative state. So endospores are metabolically inactive, and this is particularly found in phylum Firmicutes, example Bacillus and Clostridium species. At the molecular level, sporulation is governed by a cascade of tightly controlled genetic events involving the activation of master regulatory proteins, SPO0A, and temporal expression of specific sigma factors. What are those? Those are F, G, and K. These orchestrates the spatial and temporal regulation of genes responsible for asymmetric division, spore maturation, and development of spore-specific structures like the spore coat, cortex, and core. These asymmetric divisions, spore maturation, those are the stages of multi-stage differentiation. I told in the previous definition. So, this is the processes of the differentiation, total differentiation process. These are the parts, these are the steps. The process culminates in the release of a metabolically inert, structurally fortified endospore capable of surviving extreme environmental insults such as desiccation, heat, radiation, and chemical exposure until favorable conditions trigger the opposite process of sporulation, that is the germination and resumption of vegetative growth. So whenever there is favorable condition, the endospore can revert itself back to the vegetative form, that is the germination. So what are the spore forming bacteria? Say it is the bacteria's doomsday mode. It is the bacterial doomsday mode. Endospore activated. There is indestructible environment, radiation exposure, different chemicals which are harmful to the bacteria, heat waves, chemical clouds, food deprivation. Then it encloses itself as the endospore. So what are the spore forming bacteria? Gram-positive spore formers like Bacillus species, Bacillus anthracis, which causes anthrax, Bacillus cereus, also, as this is associated with food poisoning. Bacillus subtilis, this is a model organism for studying sporulation. And again, Clostridium species, those are the phylum Firmicutes. Those are included in phylum Firmicutes. Kingdom, phylum, class, order, family, genus, species. There's nomenclature of bacteria. Clostridium species. Example, Clostridium botulinum, which produces botulinum toxin, hence causes botulism. Clostridium tetani causes tetanus. Clostridium difficile causes severe diarrhea, colitis, antibiotic-associated diarrhea. And Clostridium perfringens associated with gas, gangrene, and food poisoning. Now, what are the features of spore-forming bacteria? Not all bacteria are spore formers. So, features of spore-forming bacteria, they are found in soil, water, and decaying organic matter. They are resistant to heat, highly resistant from UV radiation, desiccation, disinfectants, and they play roles in diseases, biodegradation, and industrial processes. We will come to those points shortly. Now, from vegetative growth in harsh environmental conditions, they form endospores that by the process of sporulation. So now the next slide is on endospores. Endospores are highly resistant dormant structures we all know by now, formed by certain bacteria to survive extreme environmental conditions. So what are the key points in short? Virulence factors, botulinum toxin, tetanus toxin, anthrax toxins produced by endospore forming bacteria. Endospores germinate in favorable host conditions, germination, leading to toxin production and disease manifestation. And Schaefer-Fulton stain used to visualize endospores, that is a green stain for spores and red for the vegetative cells. PCR and qPCR help detect virulence genes and genes related to endospore formation, SPO0A, SPO0F, those are the genes. Now, structure of endospore. This is very important. Complex structure enables their extreme resistance and dormancy. The structure itself prevents it from destruction. The structure itself is the cause of resistance. So first, there is exosporium. Exosporium is the thin, loose outermost layer of glycoproteins. See, here is the picture. The brown outermost side, this brown outermost portion, yellow ochre it is basically. This is the exosporium. Thin, loose outermost layer. It is made up of glycoproteins. This is the hydrophobic barrier against chemical agents. This is hydrophobic. So it prevents attraction to water. It repels water molecules. So this is the barrier against chemical agents, and variation found in some endospores like Bacillus cereus but absent in others like Clostridium difficile. Now for spore, see the picture here. Spore coats. Spore coats composed of cross-linked protein layers rich in cysteine. Those are the disulfide. Those are connected with disulfide bonds. So inside the exosporium, there is the spore coat, which is formed of basically cross-linked proteins. Proteins means cysteine mainly. So what are the functions? Enzyme resistance, chemical resistance, those are the functions. Chemical enzyme resistance protects from protease and lysozymes, so prevents destruction. And chemical resistance limits permeability to toxic agents again. So it is actually the bulletproof armor protecting them from heat, UV radiation, chemical attacks, just like prepper's survival gear. This is the spore coat. Now, spore coat, we have completed. The next point is cortex. Cortex, this is made up of peptidoglycan, but this is less cross-linked. than the vegetative cell. Peptidoglycan is also present in vegetative cell. We have studied in bacterial cell wall, but here is also peptidoglycan present. The inner side of spore coat is the cortex, but here they less cross-linked. So it maintains low water content, collapses during germination to support vegetative cell growth. Core, this is the low water activity. This is approximately 30% compared to vegetative cell. Whether in vegetative cell, the water content is 70%. Here it is 30%. It contains chromosomal DNA stabilized by small acid-soluble proteins, that is the SASPs. Ribosomes in inactive state, those are present in the core itself, and DP-cholic acid, the most important structure which is related with calcium ions, which makes approximately 10% of the dry weight of the endospore. That's like their supply of canned food. It keeps everything stable inside the bunker, which is present in the core itself. DP-cholic acid, DPA, chelated calcium ions, 10% of dry weight. It is like the supply of canned food. It keeps everything stable inside the bunker. It also resists destruction, heat destruction of the endospore. Now, sporulation as a game of survivor. In the microbial game of survivor, bacteria face harsh conditions like starvation, heat, chemicals. Endospore forming bacteria vote out their vegetative cell life and focus on building their indestructible spore to win the game of survival. Everywhere there is the survival of the fittest, the war of the survival. So those endospore forming bacteria, we have got it in the list, the Bacillus species, Clostridium species under the phylum Firmicutes. They vote out their vegetative cell life and focus on building indestructible spore. Thus, they win the game of survival. Those bacteria who cannot form endospores, they will die out due to the harsh conditions. The core is like the immunity idol, protected at all costs with DPA and SASPs. Okay. Now, what is the resistance mechanisms of the endospore? By now, we know about its structure and functions of every structure, which protects it from heat, which structure protects it from radiation, which structure protects it from chemical agents, and the cross-linking where there is presence of glycoproteins, where peptidoglycans, exosporium, basically hydrophobic, so they protect from chemical agents, the outermost structure. Then coming to the inner to exosporium is the spore coat, which can protect the bacteria from enzymes like protease and lysozymes, and chemical resistance also by limiting permeability to toxic agents. And again, the structure is made such that they can resist against harsh conditions. The cortex maintains low water content. So the sterilization processes, which uses the wet sterilization processes like autoclave, and those cannot destruct the spores easily because it maintains a low water content in the cortex as well as in the core. And in the core, the DP-cholic acid, it keeps everything stable, and also it supplies the canned food, it supplies the food in unstable conditions. So now coming to the resistance mechanisms. Arises from a combination of structural and chemical adaptations. Heat resistance, low water content dehydrates the core to protect cellular components from heat damage by preventing heat-induced protein denaturation. Because if there is higher water content, heat penetration is better. If less water content, heat penetration is much less. So it can prevent heat-induced protein denaturation as penetration is less due to low water content. This is the mechanism. Now DPA, that is a DP-cholic acid, which calcium complex stabilizes DP-cholic acid chelated with calcium. So it is a DP-cholic acid calcium complex, DPA calcium complex, which stabilizes the DNA proteins and membranes by binding to free water molecules, reducing reactivity. It binds with the free water molecules, and so it prevent the reactivity of water molecules to the heat, the response of the water molecules to heat. Spore proteins such as SASPs bind DNA, reducing its susceptibility to depurination at high temperature. spore proteins like SASP, they bind with the DNA, and this binding reduces the susceptibility of the DNA from depurination at high temperature. Now, radiation resistance, this SASP, as it binds tightly with the DNA, reducing double strand breaks from UV and ionizing radiation. Similarly, as it reduces the susceptibility of the DNA to UV radiation. Chemical resistance, spore coat. SASP full form is small acid soluble spore proteins. Small acid soluble spore proteins which binds with the DNA. Now, chemical resistance, spore coat blocks harmful agents. Spore coat is resistant to chemical substances as well as enzymes. So it blocks the harmful agents like disinfectants, oxidants, and enzymes. Inner membrane acts as an impermeable barrier. And desiccation resistance core dehydration limits oxidative damage. Stabilization of enzymes and DNA by SASP, small acid soluble spore protein, and DPA, dipicorinic acid, ensures functionality upon rehydration. Dormancy is another cause, and extremely low metabolic activity minimizes the risk of self-damage over long periods, because if there is metabolism, the metabolites produced inside the bacterial cell itself damages the bacteria. So that is the self-damage. Here, as the metabolic activity is low, the risk of self-damage is less over long period of time. So it can survive over long periods. So endospores are literally the terminator of bacteria because it is resistant to heat, resistant to radiation, and resistant to chemicals. Means it says to the chemicals, I will be back after disinfectants. And it says to radiation, come with me if you want to survive UV. And resistant to heat, hasta la vista, means safe from boiling water also. Now, coming to the process of sporulation. See, this will be easier if you go through this picture. This is the vegetative cell, and then it is a signal induction is there first. Nutritional stress such as carbon nitrogen starvation activates SPO0A. Two component signaling systems like kin A, SPO0, SPO0B, phosphorylate SPO0A. So these are the genes. After phosphorylation, they becomes activated. Now, stage 2 is the asymmetric division. Cell division starts as the genes get activated. Cell divides asymmetrically, smaller cells, larger cells, forming a larger mother and small spore. Now, engulfment. Mother cell, this is the mother cell, engulfs the phosphor, forming a double membrane structure. Double membrane structure is formed. Now, SPO0A, which stands for stage 0 sporulation protein A. When it is in stage 0, that is in vegetative cell. So, it is a stage 0. So, sporulation protein stage 0A. Sporulation protein stage 0F and sporulation protein stage 0B. Those are the full forms. We will come to the notes after discussing this picture. Now engulfs it, and then sigma E regulates gene expression in the mother cells. Now then there will be the cortex formation. Thick peptidoglycan layer forms in between the phosphor membranes. Enzymes modify peptidoglycan structures to reduce the cross-linking. And stage 5, it's the asymmetric division. And then this is the cross-linking and cortex formation all. Now, spore coat formation. Sigma K activates the synthesis of protective protein layer in the spore coat. Then is the DNA synthesis, maturation, and finally, the release of the spore. So this is a highly coordinated genetic and biochemical cascade. Initiation, stress and followed by signal induction. Those are the stresses: carbon nitrogen starvation and sporulation protein 0 stage A activated, two component signaling system phosphorylates this SPO0A. As it is phosphorylated, it starts asymmetric division. Still divides asymmetrically a larger mother cell and a smaller phosphor. Then engulfment. Mother cell, inside the mother cell, smaller force, mother cell engulfs a phosphor, forming a double membrane structure. Now, sigma E regulates the gene expression in the mother cell. Now, thick peptidoglycan layer forms between the phosphor membrane. Inside the mother cell, engulfment of the phosphor and septum formation and phosphor membrane formation. In between phosphor membranes, thick peptidoglycan layer formation. Enzymes modify peptidoglycan structure to reduce the cross-linking. Next is the spore coat formation. Sigma K activates the synthesis of protective protein layers in the spore coat. So first core formation, the cortex, then spore coat, inner to outer. Maturation, DPA synthesis and uptake occur, followed by core rehydration. Spore resistance is fully developed at this stage. Core dehydration. The mother cell undergoes lysis, releasing the mature endospore into the environment. This is the sporulation process. So that's all for today. And shortly, I will discuss the remaining part of the sporulation. Those are the germination processes and the importance of spore in microbiology, etc. The germination process in details, molecular regulation, clinical and environmental importances, laboratory techniques, and all. We will discuss those later on. So that's all for today. Thank you. Please like, share, and subscribe the channel, and leave comments in comment boxes. And there are quizzes also once I finish the topic. Thank you. Have a good day.